Hello, good afternoon, Zoology 2700. What we're going to do today, this is kind of the 5 or 10 minute flipped introduction to some of the protozoan diversity that you're going to explore this term. We're going to talk about Ciliafriends and Apicomplexans, and then on Wednesday we're going to dive into one particular group of Apicomplexans. But to remind you, so we're in the alveolites, so this group that's unified by the presence of alveoli. We've got the dinoflagellates that we spent a lot of time on Monday talking about, with red tides, and Noctiluca, the bioluminescent group, or an exemplar group that bioluminesces, as well as the Symbiodinium, the zooanthely that lives mutualistically, symbiotically with corals. Today we're going to talk about ciliates really briefly, and then start to introduce you to the Apicomplexans, and then finish that up on Wednesday. So the Ciliafriends, this group is one that is derived, the word is derived from cilia, which is Latin for eyelash, and it talks about the motion. So this group, very diverse group, but it tends to use multiple forms of cilia to move around. There are mitochondria with particular tubular cristae that form an apomorphia for this group. There's two types of nuclei, and let's talk more about the cilia, because that's really an important kind of breaking down for this group. Now you'll see in the Brusca page reference there, we're still in chapter 3, but chapter 4 in Brusca talks a lot about generalized things about surface area to volume and mobility. So lots of the figures that you're seeing here are actually going to be from that part of the textbook as well. So we're breaking a little bit of chapter 3, and then you can kind of sample or graze across chapter 4 as well. So on the left-hand side of the screen, you can see a breakdown of one particular cilia. So Cilia and Flagella, we see one cilium, that is, and its basal body with the associated fibers. And so this basal body has 9 triplets and a doublet, which is basically a way to describe how it's anchored within the cell wall. And then you see the microtubules, as look at that blown out cross section, that expand from each, expand across the length of that cilia. So the cilia is composed, you're looking now at an older figure. This is one that's from Rupert, Fox, and Barnes and isn't in your in the Brusca test. So I love this view. It's a particularly clear view of it. But there's 3 words here, nided, needy, and nitodesma, that refers to individual cilium, or rows of nideds, or rows of cilia. And then at the bottom, with the waves and the power and the recovery strokes, we'll talk about that. So let's talk about morphologically first. We've got is an individual nided. So that's in blue, that shape there, where it's drawn out that we've got a body that supports the cilium hair projecting out of it into the rest of the world. Now there, when we group together many nideds along the body, we see the row of nideds, or niti of nitids, and then the nitodesma, that underlying bundling of basal body fibers that allow each one of those nitids and the rows of niti to be firing in a particular wave-like format. And that's particularly important because you can imagine if you were, picture the individual cilium projecting out, and in your mind replace those with paddles in a canoe. And you're sitting in a canoe, and each individual of you is a nitid, and then the whole class is niti. And if you're all paddling at different rates, all of your paddles, all the blades of those paddles are going to be crashing in together, and the boat itself is not going to go anywhere. And so the nitodesma is critical towards coordinating the strokes of those niti, the nitids across the niti, so that they have these hydrodynamic effect, whereas you watch that video at the bottom there, going from right to left on the screen, that you've got power strokes and then a recovery stroke, and power stroke and a recovery stroke, where it pushes and then it recovers up the length of the body and it pushes again. And working in the opposite direction, you can see a figure from Brusca that delineates that power and recovery. Power and recovery. So both of those kind of pushing towards the center of that internal slide, of the center of the slide. Rupert, Fox, and Barnes, before they died, they talked about, there's different parts in that text that I really enjoyed because they waxed poetic about the diversity of invertebrate life. So they talked about these metachronal waves as being those that pass over the surface of the cell as wind passes in waves over a field of wheat. That's not so bad when you're talking about little Ciliafriends. You can read more about this on the 89th page in your Brusca text. Now, sometimes those cilia are short ones and they're fused. And that allows what appears, so instead of that kind of metachronal waves that allow you to paddle through, these fused cilia, called cirri, allow the Ciliafriend to appear to walk or to kind of peg its way through. And so that's what you're looking at here. The Euplodes in that video was walking across the slide. So you can see in the diagram, fused cilia. And you imagine energetically why this might be advantageous. It might allow simplified signals to be sent, because instead of thousands, you're dealing with hundreds or dozens of individual projections out into the world that you'd have to coordinate. And for a simple organism, simpler is probably more advantageous. So here what you're looking at is cilia. Focus on the center of the cell and you see almost what looks like a vortex or a whirlpool into the center. This is an individual paramecium. So this is a Ciliafriend that you're probably familiar with. And you can see little other protozoans and little algae and kind of euglenids maybe beating around in that water, surrounding the larger paramecium. And if they come close, there's another one kind of moving through, but you can see that mouth-like, that's setting up a feeding current to bring food in small projectiles inside or close to that cell wall where they can be engorged and consumed. So kind of complex. We're getting into moderately complex behaviors and biologies here. Now, if you're interested in ecology, or if you've taken ecology or you're taking ecology and you heard about Gaussian things. Now, Gregor Gauss was a Russian ecologist who talked a lot about, and who we still use to understand competitive exclusion via inter-specific competition. And all of those experiments where the carrying capacity of one particular species is affected by that of the other when they're competing for the same resource, all of those experiments that he did in the 30s were done with different species of Paramecium. So these are organisms that he took advantage of their simplicity and their size so that he could set up these large experiments of relatively simple communities, one or two species, and then looked at who was eating what, when, and where, and how one species affected the other. So that's our zoom through the Ciliafriends and the cilia. And now I'm going to give you a quick intro to this last remaining group of the aviolites, and that's the Apicomplexans. Now we're going to unpack this group a lot more, particular branch of it on Wednesday. But I wanted to tell you about a little bit of what characterizes this group right away. So it's moderately diverse, about 5,000 species, and there's important parasites within this group. So malaria, right away, coccidosis. So this is a group that affects us, and our understanding of it affects how well we can live and where we can live and how well we're doing in different parts of the world. So the apical complex, you can see right up there on the top. This is, I'm going to show you a different diagram of this in a second, but this is an apomorphy for the group. So all of these parasitic apicomplexans have this apical complex where you see there's these conids and micronemes projecting up to the front, and then a polar ring right at the very top. They have no cilia, so that is an easy way to differentiate them from the Ciliafriends. And that apical complex tends to be used by the individual apicomplexian to attach or penetrate to host cells. Now they often move, it says, by gliding motion, and then underneath it I've written that they don't have cilia, and that's to remind me, to remind you, or to let you know that there's a whole bunch of how they actually are moving that we don't know. This is one group where we tend not to be able to culture them. They live parasitically, and so sometimes those intra-host movements inside the host we know very little about because we can't observe them. So the apicomplexa with this apical complex, and you can see another group of it here, another in the scanning electron micrograph here. So Plasmodium, the group of apicomplexans that causes malaria, and Toxoplasmosa, which causes toxoplasmosis in humans, and that's an example that we're gonna unpack on Wednesday. Let's take a look at these pre-connoital rings, the conids, and the plasma membrane. So these microtubules, this is what we're called micronemes in the other slide. So down here you can see them projecting up towards the polar ring, and that's essentially what you're looking at is a complicated, almost concentration of intracellular bodies or structures that are adaptations towards a parasitic style of life, towards penetrating and or entering the cell body of the host. So with Plasmodium, which is the most famous example of the apicomplexa, the parasite that causes malaria. So it's vectored by the Anopheles mosquito, and millions of individuals infected every year. Of those, a relatively small proportion of them die, but if malaria yourself, or if you know someone who has had, you know that the effects are debilitating and reoccurring. And so the effect has an enormous economic effect on a region where malarial outbreaks happen, because those that are struck by it are laid out for a long periods of time. A severe attack can cause the loss of 1/3 to a half the total number of red body cells. So you're just not moving oxygen very efficiently at all. And you've run into malaria as well if you're thinking about human evolution, about sickle cell anemia, where the red blood cells in some lineages, human lineages, are abnormally shaped or differently shaped, not like a big donut, but more sickle or sickle shaped. These are adaptations against malaria. And you can see the diagram of the complex life history there. Not worried about too many of the details, but you can consult figure 3.16 there if you're interested. And we just came in from my oldest son and I skating in the middle of his kindergarten day, and so that makes me think about malaria, because if we've been in Ottawa, some of you are in Ottawa, and perhaps the canal has opened this COVID year. I'm not sure if it's a great idea, but what makes me link in my brain malaria to the Rideau Canal, and that's the fact that in any particular year, as that canal was being constructed, 60% of the workers had malaria, predominantly at that time Irish workers that had that disease. And so here's a diary entry and one of the historical markers along the length of the canal. So a diary entry from the Reverend William Bell of Perth that stated, repeated attacks of ague have left me reduced in body and discouraged in mind. This was malaria that he was affected by. So it was by far the largest cause of disease in the important construction in eastern Ontario of the Rideau Canal. Now the last bit of pre-lecture apicomplex stuff to think about is a link to the end of today's lecture when we talked about coral. Now, we kind of had our minds exploded a couple of years ago, those of us that think about these things, because Patrick Kwong and co-authors from Patrick Keeling's lab at the University of British Columbia had a paper that came out in Nature where they talked about the fact that the symbiosis in coral that historically we had thought of as a cnidarian hosting a dinoflagellate that was photosynthesizing in the Symbiodinium group. And what they used in terms of scanning electron microscopy and sequencing and a couple of other high imaging techniques was to demonstrate that in fact this partnership is not just of two, it has a third partner. And the third partner is otherwise apicomplexan. So historically apicomplexans likely started out as a photosynthetic group that secondarily adapted towards becoming a parasite. And in most coral lineages now, we've seen evidence that there are apicomplexans that are hiding very cryptically within and producing. They're integrated into the byproducts of the coral of its coral host, and so this famous example of mutualism that we all know about, we've been missing this third partner. This is a mutualistic relationship, not of two, but of three. So I'm going to leave you to watch this video later on if you want, this little Twitter post they're very simple, kind of couple of minutes. And I'm going to thank you for joining this kind of flipped part, and we'll see you on Wednesday.
Coral reefs are among the most biodiverse and vibrant ecosystems on Earth. They are possible because of tiny symbiotic algae called Symbiodinium that live inside coral tissue, allowing corals to capture the sun's energy using photosynthesis. Now, scientists have discovered that another symbiont is also present, hiding in most corals, including stony corals, soft corals, anemones, black corals, zoanthids, and mushroom corals. Called Corallicolids, these microbes are oddly related to parasites that cause diseases like malaria and toxoplasmosis. Stranger still, deep inside their DNA were discovered genes for making chlorophyll, the essential light-capturing molecule that enables photosynthesis. However, unlike Symbiodinium, Corallicolids were missing other important photosynthetic components, suggesting that they are using chlorophyll in a new and unusual way. As such, Corallicolids likely resemble an evolutionary intermediate between photosynthetic algae and parasites. It's unclear how the mysterious Corallicolids interact with their coral hosts and how they may affect reef health. But it is a piece of the puzzle that is finally getting its day in the sun.